everyone, and welcome to another episode of GenLive. I'm Juliana Lemire, science writer at Gen, and I'll be your host today. On today's show, we'll be discussing cancer research with two experts, Sigal Kadak from Harvard Medical School and the founder of Foghorn Therapeutics, and Tobaloba Oni, a newly appointed Whitehead Fellow at the Whitehead Institute. We'll hear from them in just a moment. I'm joined by two of my colleagues today, Alex Philippidis, a GenLive regular, and Anjali Sarkar, an editor at Gen, whom we're welcoming for the first time. Hi, Alex. Welcome, Anjali.
Howdy,
Hello.
Juliana. Hi,
Hello.
Anjali.
Also, today's episode is sponsored by Isoplexis. Isoplexis is a life science technology company building solutions to accelerate the development of curative medicines and personalized therapeutics. Their award-winning single-cell proteomic systems reveal unique biological activity in small subsets of cells, allowing researchers to connect more directly to in vivo biology and develop more precise and personalized therapies. In a recent study published in Nature Medicine, researchers used Isoplexis' platform in a phase 1 trial of TIL treatment for metastatic lung cancer, demonstrating the power of Isoplexis' single-cell functioning phenotyping. In the study, polyfunctional strength index, an indicator of the number of cells capable of secreting multiple types of cytokines, reflecting the ability of a T cell to carry out multiple functions, was used to measure the ability of single T cells to secrete multiple different cytokines after stimulation. This is just one of many examples of Isoplexis' unique ability to provide predictive quality metrics. The Isoplexis platform fills a critical need in the field, accelerating next-generation therapies. So, despite a budget of more than $6 billion allocated to the National Cancer Institute and a highly publicized cancer moonshot, the development of new cancer treatments remains a daunting challenge. Some of the promising new frontiers in cancer drug development focus on areas such as immune modulation, epigenetics, and even harnessing the microbiome. The promise of the next big treatment breakthrough lies just around the corner. Today, we'll discuss where cancer research is now, the challenges it faces as it moves forward, and the innovative new ways that cancer is being targeted to develop future therapies. We're joined by two very special guests today. Sigal Kadak did her graduate work at Stanford. It was there that Sigal first made the link between mutations in the BAF protein complex and human cancer. This work launched her research studying chromatin regulators and developing therapeutic approaches for cancers based on targeting mutated BAFs. Not only does she run a research program at Harvard Medical School focused on this work, she also founded the company Foghorn Therapeutics to develop drugs in this area, and we're excited to learn more about all of it. Thank you so much for joining us today, Sigal.
Thank you so much for having me. It's really a privilege to be here.
Just down the road from Sigal in Cambridge, Massachusetts, Toby Oni is just starting his lab at the Whitehead Institute. Toby was named a Whitehead Fellow earlier this year in February. For those that don't know, the Fellows Program provides early career scientists an opportunity to set up research programs of their own as an alternative to traditional postdocs. Recent PhD and MD graduates with a stellar record of research are given the resources to work as principal investigators. And there are big footsteps to follow in this program, including those of Eric Lander, the late Angelica Aman, and more. Toby's work focuses on new methods of understanding, detecting, and potentially treating pancreatic cancer, something we'll learn more about today. Hi, Toby. Thanks for joining us.
Hi, thanks for the invitation. Looking forward to the discussion.
And we would also like to answer your questions too. So please put any questions that you have for our guests in the Q&A section of the Zoom, not the chat. Okay, so we'd like to start today by asking both of you some big picture questions. So Sigal, I'll start with you.
Sure.
If we were to ask you to give us a state of the field for cancer research, what stands out to you? What is the state of cancer research today?
It's a great question. So thanks so much again for having me. I think, something that comes to mind that's part of our daily routine here in the lab and our daily thinking about, in our case, chromatin and gene regulation really comes from this idea that, sequencing studies performed in the context of human cancer and other diseases have really provided the blueprint, if you will, for the perturbations that drive disease. And it's really over the past near decade or so now that we've seen the wave of studies presented across disease types that really indicate for the first time what are the genetic determinants of those cancers or other diseases. And our lab continuously uses the insights gleaned from large genetic studies such as TCGA, the Cancer Genome Atlas, and others to inform the types of mechanistic deep dives that we do. I always say that we're at ground zero because of the sequencing. Sequencing was a very powerful, new technology that can be readily applied to essentially every tumor that walks into every patient that walks into Dana-Farber now gets sequenced. And it's so powerful, but it also sets us as researchers back at ground zero, trying to really understand what were the key genetic drivers of these diseases, sometimes multiple drivers, and that's where I see so much of our attention. If you go and look at the types of work being done by so many, it's really centered on the unexpected findings that came from that genomic sequencing era. And the other, in parallel to just sequencing of human tumors, the other things that genetic studies allowed, for example, massively parallel determination of dependencies, key targets that are vulnerabilities in certain cancers that are based on their genetic makeup. And that's, really provided an understanding of the key targets in this area, certainly in chromatin regulation. I think Toby's going to talk to you a lot about some of the targets that antibody-based therapies can affect. And, I think that is really where we are right now. And, you just see new results even just this past weekend from ESMO and, these larger clinical oncology meetings where they're presenting the results of some of this. It's to see, on target specific inhibitors for a specific mutation. In the case of, ESMO, Mirati's, KRAS G12C mutation, covalent inhibitor directly targeting this. So you really are starting to see the advent and development of very on-target mutationally defined rather than just tumor type defined therapeutics. And I think that's a very exciting, new wave that we're all on right now.
Hey, terrific. Toby, do you have anything to add about the state today of cancer research?
Yeah, I mean, and this actually complements what Sigal just said. So kind of thinking beyond cell autonomous requirements of these mutations, what we're starting to really appreciate and understand is the idea that cancer cells don't live alone. We know this, but actually for a long time, we've really studied cancer cells in a dish just by themselves. And so the paradigm we're operating under now is the idea that the environment, the ecosystem that cancer cells live in matter a lot. And this actually can define some of the responses to therapies. And another consequence of this is the power of the immune system and the ability to harness the immune system to fight cancer. That is super important these days. And I think, the initial studies of immune checkpoints in melanoma really showed us that when you use the immune system to attack cancer, there's a property that you constantly get that you typically don't get with other forms of therapy, which is durability. You see a lot of these patients living for a very long period of time, and they're basically cured in some cases. And that's because the immune system has memory. And so if the antigen comes back, if the cancer comes back, the immune system can really, basically eliminate that cancer again. So I think those two aspects, it's really the non-cell autonomous aspects that we're trying to really push on these days.
I think picking up on that, I totally agree. And so Toby and I are kind of at two different sides of the spectrum of cancer research. One, very cell intrinsic, which is our work, and very cell extrinsic microenvironment, which is Toby's work, each of which is extremely important, in our effort to combat cancer. I think that for both of, what we said about the different areas here within cancer research, both of these explanations begin to define sort of what would arguably often be called sort of a bit of a failure or, lackluster results of the microarray era, where we were just looking for expression of certain proteins, despite very effective and, in some cases, small molecule therapeutics targeting a specific pathway or specific protein, a lot of times this was insufficient. When you go look at the clinical responses or even model systems, it was insufficient to really achieve a response, and especially, as Toby's mentioning, a durable response, in these patients. And I think so much of that comes from the fact that at the time, we didn't know the genetic underpinnings of these diseases, and we didn't know the repertoire of immune cells and immune factors that could be contributing to these responses. And I think now we're in a very exciting time where both of those are possible. And I see a world going forward where certainly combinations exist, where, there are specific inhibitors, say targeting the chromatin regulatory system in our case, paired with immunomodulators, antibody-based therapies and others, right, as very effective combinations for cancer patients.
So yeah, picking up on what you were just saying, I mean, it's so why is cancer treatment so complicated, and where you would need, multi-pronged approaches and targeting different aspects of it? Like, what is it about cancer that leads to the necessity of that approach?
And I think so much of this comes back to this idea, again, that we're on many cancer types, we're at ground zero. We don't yet understand the biologic mechanisms that are truly contributing to the development and then ultimately the maintenance and often metastasis of these tumors. I think, from our perspective and the strategy that our lab has taken and subsequently Foghorn has taken is just to understand the basic biology at its most fundamental levels. In the case of our research, our laboratory studies large macromolecular machines in the nucleus of our cells. In each of our trillions of cells, there's 2 meters of DNA that must be compressed and condensed inside to fit the nucleus of a cell that's less than the size of a pinhead, less than one can see by the naked eye. And of course, that means there's a lot of fold compression, there's a lot of packing. So we study the unpackers, those that would be needed to unpack that DNA and allow accessibility of certain segments of DNA to be transcribed into genes and certain genes to be turned on. You can imagine, just like in an orchestra, one off note or one note that isn't played can result in an entire piece being off course. And that's really what we see in a lot of these tumors that have defects in the chromatin regulatory system, or in the case of what we study specifically, the case of these machines that regulate genomic accessibility, regulate which genes are turned on and off at the right times. That sequence and timing is so critical. You see programs, whole musical pieces, if you will, whole programs of genes that are now affected, not just one pathway or another. And so this gives rise to certainly one aspect of the answer to your question, which is why is it so complex? It's numerous pathways that are affected. And I think, Toby will have a lot to say about not only, again, the cell intrinsic effect, but what's happening in that microenvironment.
Yeah, I mean, absolutely. I love that you really paint that picture of the plasticity, which is really based on this like complex, gene expression programs. And one thing we understand is that, cancer is really, it evolves and it's dynamic and in response to treatment. And then you have escapees. And the selective pressure of any drug will drive that evolution towards some other, types of the tumor that doesn't respond. Another aspect of this is actually heterogeneity. So, the way we're starting to understand this is that, even within the tumor, you have, the tumors have evolved differently, both genetically and in terms of epigenetics. So not even within the same cell, but within the same tumor, you have different tumor cells or different clusters of tumor cells that behave differently. So, when you put that selective pressure of a drug, then, maybe you kill certain, subclones, but then some clones are still there. And one aspect of this really is the way we try to study this. And this is important. I see cancer research as kind of like a modeling business. We're all in the modeling business. Our discoveries are as good as our models. And if you study pancreas cancer, for example, the best model that exists out there is, the KPC model, which incorporates KRAS mutation and p53 mutations. That is great. It actually models the human disease very well. But guess what? You look in the patient, they have SMAD4 mutations. They have, ARID1A mutations. They have all sorts of other mutations that we don't even incorporate in our models. And so I think starting to develop models and starting to actually, answer questions in different models that recapitulate that patient heterogeneity. And actually, one aspect of this is the organoid models that we really helped develop in the past couple of years, particularly for the pancreas, and which allows us to move a little away, or actually, complements the 2D cancer cell lines that we've been working with traditionally, and allows us to really, look at different types of cells. And account for heterogeneities even from patient-derived samples. And I think that aspect of heterogeneity really accounts for one of the reasons why it's so hard to treat cancer.
Picking up on your comment of the model systems, I think that's this is a critical determinant, right, of our, success and our ability to understand mechanism. And I think, for us, at least the way we have the perspective on the chromatin regulatory system, specifically ATP-dependent chromatin remodeling complexes, is that so little was known, but yet this was catapulted to the forefront of attention, just given the genetic sequencing results that these are mutated in over 20% of human cancers, and by a number of different mechanisms that our group and others have worked out are involved in so many disease processes. This has really brought them to the forefront of attention by both the academic and pharmaceutical industry. Yet you realize, you're ready to prosecute those targets, yet you don't understand the basic biology. And so on our side, before we can even get to the model systems, I always view it, and I hope that we have a chance to collaborate in many cases down the line, I think we really argued that we had to take a couple steps back almost before we can take meaningful steps forward. Sometimes you really have to understand where you are to be able to take the most meaningful steps forward in terms of taking this to a more translational level. So for us, that was a lot of basic biochemistry. These components of these 11 to 15 or so subunit entity complexes are perturbed in different cancers and perturbed in different ways, either by a complete deletion, the whole puzzle piece of this is missing, or there's a single residue that's mutated that affects the activity of this remodeler complex, and so on and so forth. But there was no knowledge as to how this thing was pieced together in a cell. How does the cell put together multiple pieces to fit into one cohesive complex that's working together in a coordinated series of actions on chromatin to enable DNA accessibility? How does it come together? What's needed for what? What's the order of assembly? What are the determinants of its activity? All of these are critical questions that went unanswered. And so we spent the past, 5+ years trying to work out as many of those, what we thought were most critical entry points, if you will, to being able to now take this in a translational vein and drive this forward. And I think now we're in a position, excitingly, where we really can use the model systems, develop model systems that can faithfully recapitulate the human cell biology, and have that aid in our goals to try to develop targeted therapeutics.
Okay, terrific. Thanks so much. We're going to dive deeper into all of this now. And I just want to remind our audience, please put your questions for Toby and Sigal in the Q&A section of the Zoom. And so now I'll pass it over to my colleague, Anjali.
Hello. So my question is for you, Toby. Taking a step back, can you tell us a bit about how you came to be interested in pancreatic cancer research during your graduate studies?
Thank you for the question. During my graduate studies, when I started, I really wanted to work on a challenging problem. And, they're not, when you look at the cancers that afflict humans, pancreas cancer is one of the worst out there. So this was, in hindsight, maybe if I wanted to have a more fun social grad school career, maybe I should have chosen another problem. But I chose pancreas cancer, and I went to David Tubison's lab in Coastman Harbor Laboratory. It developed the KPC model that I spoke about earlier, and it was starting to really think about newer models. And at the time when I joined the lab, I helped develop the organoid model, which now is widely used in the field. And the beauty about these organoid models is that it allowed us to really model the histopathology features of human PDAC in mice. And that allowed us to now, put the cells in their natural environment recapitulate this, microenvironment and start asking important questions. And importantly, it allowed us to develop, antibody-based reagents to really, address critical aspects of pancreas cancer, such as early detection, being able to detect it earlier, and also just, being able to direct drugs to these tumors. And so being able to actually answer these questions at the end of my PhD was probably one of the most satisfying things that I've done. But, leading up to that, I really wanted to, address a challenging question, and pancreas cancer was that question.
And then you received the Whitehead Fellowship. How does it feel to be a Whitehead Fellow right now?
Yeah, it's terrific. I mean, it's great. Day one, I came in and I had an empty lab, and I was like, "Oh, this is mine now." But beyond that, I think what is really special about this is that you're given a framework, you're given a support system, you're given funding to really take the most risky project and just do them without the pressure of teaching and doing other things, and it really inspires you to be bold. Yeah. And in this line of work, in this academic line of work, it's very tempting to try to take little increments and just survive the next grant and survive the next publication. And before you know it, 10 years is gone, and 20 years is gone, and you're stuck in that incremental leap. And this is not, a feature of individuals. I think it's more a feature of the structural framework of academia. What this position allows you to do is to go beyond that and actually take that leap. That if you fail, it's okay. But it allows you to be risky, and that's what I really love about it. And that really underlies what we're trying to do with, trying to harness the immune system to fight pancreatic cancer, and importantly, leverage post-translational modifications towards that goal.
Definitely, the luxury, the have to have that opportunity to make mistakes is definitely a very good way to be. So. You focused on pancreatic cancer, and as you mentioned, and Sigal mentioned too, is that pancreatic cancer is one of the most difficult types of cancers. It progresses quickly than other types of cancer. What is it that makes pancreatic cancer so difficult to treat, and so that it progresses so quickly before you can even detect it? What is it about pancreatic cancer that's so difficult?
That's another great question. So pancreatic cancer is really, we're still trying to understand the biology, but just from the get-go, the anatomical location is not amenable to being detected earlier, right? So the pancreas is behind the stomach, and unlike skin cancer where you can see a lesion, or breast cancer where you can feel a palpable mass. Typically, when you have pancreas cancer, and you don't know for a long time until it's too late and it's spread to other places. In fact, 80 to 85% of the patients are diagnosed at a point where you can't, it's spread to other places. And so early detection is such a big problem in this disease because of the anatomical location. The other aspect of pancreas cancer is its microenvironment. It has this really, a stroma-dense microenvironment that is filled with, extracellular matrix and many other cell types. And one way that my old mentor used to actually describe this is like an oatmeal raisin cookie, where the raisin is the cancer cells, and the rest of the oatmeal is the stroma. So you have those cancer cells in a sea of stroma. It's one of the most salient characteristics of pancreas cancer, and now we're starting to understand that this actually underlies one of the reasons why it's so chemoresistant and is so aggressive. And we're still understanding the biology of this tumor, but this is one of the reasons. And lastly, if you think about the predominant mutations or genetic alterations that really define pancreatic cancer and drive this disease. I spoke earlier about KRAS, P53, SMAD4, for example. These are, proteins that are traditionally not targetable. Now, I'm pretty sure we're going to talk about, druggability, which is something that is really changing these days. But, KRAS P53, they're not amenable to your, run-of-the-mill small molecule inhibitors, right? And so because those driver alterations are not amenable to that, it's been really hard to go after them. And so we've had to go after, accessory pathways and downstream pathways and things like that. And till today, chemotherapy is still the way to go in pancreas cancer.
We'll come back to this, but for now, I'll pass it over to you, Juliana.
Oh, sure. Okay. So, Sigal, I'm wondering, you work on chromatin remodeling in cancer. How much of the work that's going on in cancer is epigenetics based? And what have the major findings been in this area up until this point on chromatin remodeling in cancer?
This is a great question. So, we can estimate roughly that up to 50% of human cancers are driven by or have a mechanism that contributes to oncogenesis that's centered in chromatin regulation. This is a vast, highly interconnected system whereby the series and timing and order of interactions of proteins, protein complexes sitting on DNA regulatory sequences themselves, RNA modifications all work together in concert to achieve an architecture of our genomes. And that architecture is very critical for the regulation of gene expression. We study a major determinant. When we saw the sequencing studies starting to emerge, and you see many of the pathways that Toby just highlighted, signaling pathways such as RAS signaling, MAP kinase signaling, EGFR signaling. These are definitely top ranked hits in terms of sequencing and in terms of frequency of genetic perturbations in disease. And this is why there's been so much attention over signaling pathways. But in addition to those common culprits that have been longstanding targets of drug discovery efforts in both pharma and academia for many, many years, the most surprising element to come from those sequencing studies was the fact that chromatin regulatory processes are frequently perturbed. And this was really not expected. I think a lot of, the research, especially earlier research that was performed in the context of yeast and increasingly in flies and then ultimately in humans, would have predicted that these would be much more required for just homeostatic functions of cells and hence not selected for in the case of a cancer. But, genetics really turned the hypothesis upside down and showed us that unexpectedly, some of the most frequently mutated genes are in these categories of chromatin regulation. And there are a lot of different types of proteins. There are transcription factors. There are, DNA methyltransferases. There are, proteins that perform and protein complexes that perform histone modifications. And then there are these ATP-dependent chromatin remodelers, BAF complexes, for example, that we work on. And we were just struck to see that it's so frequently mutated that it's called its own process now. It's called its own category, even though these BAF complexes are just one of 29 different families of remodeling complexes, all of which have some catalytic energy to be able to rewire, if you will, or reorganize the architecture of the genome and its accessibility. And so clearly this was a place to focus. Genetics doesn't lie. It tells you exactly where to look. And so for us biologists trying to study this was a key entry point by which we could then go and go in for several deep dives on specific cancer types, just like Toby, who really connected with this area that he's now dedicating his life to, this really came from my graduate studies as well. So both Toby and I, had these experiences during graduate school and findings during graduate school that sort of catalyzed a new area of exploration, I would say. And, for me, I was working as a graduate student in Jerry Crabtree's lab at Stanford. Jerry is a world-renowned developmental biologist, and at the time wasn't studying cancer at all. I came in with a real, desire to study cancer and just trying to understand a process that he had characterized so beautifully in the context of human development, whether that could be hijacked in some way in cancer. And as we all know, cancer is often development gone wrong. So thinking about it from that perspective, we are able to link a very specific cancer known as synovial sarcoma, which we still study. It's a very specific a case study really of how this remodeler can go wrong. And it's one of the reasons why we've had such extensive studies on that rare cancer. But we link that rare cancer to the aberrant activity of this large remodeling complex that sits on DNA. And essentially, in essence, what we found was that a fusion protein that is 100% the driving force in this cancer called synovial sarcoma. 100% of patients have a fusion protein. And there's a fusion protein, for those who are not familiar, are two proteins that are fused together by a problem in the DNA recombining incorrectly. And so you see two proteins that are come together because their genes are now together. So the product of those genes is now a fusion protein. One part of the fusion protein is actually a member, we discovered, of this BAF complex, is a member of the chromatin remodeler. And then the fusion piece is a totally aberrantly added little 78 amino acid tail. And in every single case that we found is that by just perturbing that subunit of this remodeler, by adding that small piece, it caused that remodeler complex to go to all the wrong sites on chromatin at all the wrong times, essentially taking what was supposed to be a nice, mature muscle cell differentiating down its lineage and actually reverting it back to a stem cell phenotype. You don't want a stem cell in your mature cell populations. And so this reversed the gene expression pattern back to essentially stem cell gene expression. And that all happens because this remodeler is opening regions of DNA that should be closed and allowing genes to be turned on, the wrong genes at the wrong time. And so we took that rare cancer and made significant headway. In fact, there's actually a clinical trial that we at Foghorn now have initiated just a couple of weeks ago in patients with the targeted therapeutic for synovial sarcoma based on these findings, which is very exciting. But in addition to that, we asked the question, where else does biology use these mechanisms? I always say biology uses mechanisms in more than one place. So if you find some exciting mechanism, it really behooves you to ask the question, where else has the cell become smart and use this type of mechanism, whether it's for disease or whether it's for a normal process. And so sure enough, we've been able to identify multiple other disease settings, particularly cancer settings, where factors that can drag or re-navigate, if you will, these complexes on our 2 meters of DNA, how they can act and how they're interacting with these remodelers. And this is providing a whole nother platform for both mechanistic discovery and drug discovery.
Oh, terrific. I know that Alex is very interested to ask more questions about Foghorn Therapeutics, but before he does, I just want to ask a more general question. Are there other attributes besides its huge role in this process that make the BAF complex a good drug target?
Sure. So I think, historically, of course, it's been a challenging target just to understand, let alone drug. These are driven by catalytic energy. So it does have an enzymatic function. Enzymes have been drugged before. So it is in essence an enzyme, but it's a multi-component enzyme with a lot of allostery. It's a huge, almost 2 megadalton complex, very, very large in size. It's about 12 times the size of a nucleosome. And so this, there are a number of aspects of it that could conceivably be druggable, although this is the first time it's being drugged, so we have yet to understand how this works. I think what makes it uniquely special is also that I say the complex, but what I actually mean by that are thousands of complexes. Even in the context of one human cell, even at the single cell level, there are hundreds of total possibilities for how these complexes can be pieced together. There are actually 29 different genes, but the complexes themselves are about 11 to 15 subunits, depending on the complex. And there's 3 sort of final forms that we've been able to work out, 3 final form assemblies, if you will, of how the pieces come together. But within each one of those, there's N choose R combinatorics. So by simple math, you can figure out that there's hundreds of possibilities. And so clearly, evolution has it such that we needed those combinations. It's all too costly for evolution to duplicate and diverge subunits without, or proteins and genes without a need. And so we have just slight nuances in function that can completely change the behavior of this remodeler complex. And that's given us as a lab, a significant number of challenges. You're working with such a heterogeneous mixture just within any one given cell, let alone the heterogeneity that's going to then extend into what Toby's talking about, the microenvironment. How are you going to see how the ramifications of that heterogeneity? So I think, that's a challenge, but it's also an opportunity. And it's an opportunity in the drug discovery side for specificity, because clearly there's a reason for specific functions that those different combinations have. And we're really eager to understand those and be able to ultimately target them.
Okay, awesome. Thanks. So before we keep going on drug discovery at Foghorn, I think we're going to go back to Anjali for a little bit and talk about pancreatic cancer.
Sure. Toby, let's talk a bit about organoids for a moment. You've used human and animal model organoids to explore pancreatic oncogenesis, and this is a new model coming up that so many different labs are using. How important is the use of organoids in pancreatic cancer research, particularly in the context of this heterogeneity and complexity that you're talking about? Can this heterogeneity really be modeled in organ systems, and if so, how?
Another great question. Yeah, so, before we had organoid systems, we had a handful of cell lines, I mean, to study pancreas cancer. And actually, when you implant these cell lines in mice, what you find is just, you just find an undifferentiated sheath of epithelial cells. That's what you find. So you basically have all raisins, no oatmeal. And so this does not really model, it doesn't allow you to even model properly, pancreas cancer. And so the organoid system, when we developed this, really allowed us to actually, within the same organoid derived from the same patient, you start to see multiple clones that you can now go back in the patient and actually see that clone. And importantly, you can actually derive these organoids from, fine needle aspiration. So you don't actually have to like cut out the tumor. You could actually just suck out a little bit of the tumor and actually, develop this organoid. And what this allows us to do, obviously, is personalized medicine. In the past, I mean, you just cannot, make 2D cells from every single patient. Actually, most of these cells fail when you try to just grow them on a plastic dish, but most of them succeed when you try to grow them as organoids. So it allows us to, number one, study inter-patient heterogeneity and also intra-tumor heterogeneity. And so that the combination of those two will allow us to actually properly determine what therapies will work across patient types. And in my case, I was able to actually find epitopes, antigens that are expressed by pancreas cancer patients that are expressed across patient samples and could reliably be biomarkers or be used for early detection. And so that's where the organoid system really helps out a lot.
Are there any drawbacks to using the organoid model that you things that you cannot do in the organoid pancreatic cancer model that you would have to defer back to using the animal models, particularly in the context of how the immune system is being studied to attack the tumor cells?
Absolutely. Autochthonous models are still very important. One thing I would say right away is that, to use the organoid models, you probably have to have a war chest. It's like they're not cheap. So in comparison to cell lines, they're very expensive. But what the organoid model allows us to do is to actually take this, cells, put them back in the pancreas and actually try to model you have immune infiltration. We've actually recently done a really nice comparison of, animal models to the autochthonous models and compared them with, organoids when we transplant them back into the pancreas. And you get the same, very similar levels of immune infiltrates. If you look at the macrophages, the monocytes that come in, the T cells that come in, which is not a lot, you see that it's very comparable. Now, what the organoid system might not allow you to do is actually to really study early disease, where you study this disease from inception, and study, for example, the roles of inflammation in allowing these tumors to grow out, and that kind of like in situ questions are harder to study with the organoid models because you always have to inject these organoids back into the pancreas. You're actually making some inflammatory perturbation by injecting them back. And you know, for example, you know, if you put back cells that only have KRAS and don't have p53, you know, a lot of them don't grow out very well. So early disease in the setting of, you know, KRAS only mutations, it looks like the autonomous models really are way better for that.
Using these organoid models, what are some of the significant translational advances that have been made in pancreatic cancer that are being pushed towards the clinic?
Yeah, so actually, currently there's a past trial being done, and it's just a personalized medicine trial. So that's the first thing right off the bat that allows you to, you know, these organoid models, you can establish it from every single patient, post-treatment, you know, pre-treatment, and actually just ask critical questions about the heterogeneity or the response to therapies. And we see that, you know, these organoids, you know, actually predict, for example, responders to chemotherapy. And there's this past trial is currently being done to actually confirm and validate this aspect. And I anticipate that these organoids will allow us to do that. So that's a current direct application. You know, in terms of research studies, I think, you know, a lot more people are using these organoid systems, and you know, it's becoming part of the pipeline in terms of biobanking of patient samples. So, you know, when you collect samples from patients, one of the things you want to do now is to actually establish organoids from there. And I anticipate that this will really be part of the clinical protocols going forward.
I guess picking up on that, Toby,
Yes.
it's so exciting. You know, it's so exciting to hear about, you know, these organoids and just their power. You know, not only for the basic mechanism again, but for drug discovery as well.
Yeah.
You know, in light of just this, I was just kind of floored by this data that came out of ESMO. I'm sure you're aware of it, by, you know, by Mirati and a little bit of Amgen as well, right? For these KRAS mutant specific covalent inhibitors.
Yeah.
You know, you'll have to remind me, you know, what percentage of pancreatic mutations have that exact mutation, but, you know, their whole concept there was that by either monotherapy, which was also effective, but particularly monotherapy combined with cetuximab, you know, Erbitux, really showed significant responses, right? This was a pretty impressive sort of 43% response.
Yeah.
I'm just curious, you know, how you think about that, in the context of the model systems, and, you know, especially in combining the results of that type of a trial with, you know, where do we land that in terms of combination with other antibody-based therapies? Because it seemed pretty promising for the first time.
Yeah, that's a great question. So unfortunately, so what's just referring, so let me go back a little. What's just referring to is this you know, new drugs that target KRAS. So I previously said KRAS is an important genetic alteration in pancreas cancer. However, the particular alteration being targeted currently is a KRAS G12C mutation.
Right.
About 1 to 3% of pancreas cancer patients have that, so it's not that much. And you know, preliminary data shows that it doesn't really respond well as a single agent.
Yep.
But when you combine it with, you know, for example, EGFR inhibitors or antibodies, then you get a response. One thing, you know, particularly that we're excited about is the ability to actually, you know, predict response, you know, based on cell autonomous adaptive mechanisms or microenvironmental adaptive mechanisms using these organoid systems. And you know, as Sigal beautifully alluded to, these models will allow us to do that more and more. Something we're super excited about is kind of the combination of KRAS inhibition with immunotherapy, for example, because we think, and we're starting to understand now, that a lot of these oncogenes not only transform cells, but they give them this, you know, ability to evade the immune system. And so by, you know, blocking this oncogenes such as KRAS, then, you know, can weaken the cells and sensitize them to immunotherapy.
Right.
Thanks, Toby. Alex, over to you.
Thanks, Anjali. And Sigal, I'd like to learn a little bit more about Foghorn. What prompted you to start a company and have one foot in industry and the other in academia?
It's a great question. So, I made my way from Stanford across the country to start my young lab here at Dana-Farber, Harvard Medical School on the Broad. And, with the mission to really, having been prompted by these exciting sequencing studies I told you about and that mechanism and that one rare cancer, we really were eager to find all the full swath of opportunities by which these complexes can maybe be perturbed in disease as a way to not only understand disease mechanisms, but also understand the true biology that underwrites all of our cells, underwrites the gene expression programs of all of our cells in the normal cell and tissue context. So that was our goal. Our lab is centered in basic mechanisms, basic science, biochemistry, biophysics, increasingly structural biology, and we layer in epigenomic and chromatin biology approaches with even systems biology and machine learning to take a really comprehensive series of approaches to understand the science. Now, as basic scientists, as I started to piece together my lab, different members thereof, we were making exciting discoveries, but at a rate that was fast and at a rate that would never be met with, in parallel development on our own of therapeutics for patients. And we knew that this is a really, this entire bucket of mutations, this entire class of BAF perturbed malignancies, which is ever extending as we continue to understand the mechanisms, is a pretty significant bucket and has major unmet medical needs that are targeting these specific perturbations in these cells. And we knew all too well, I knew all too well that I'm certainly not a chemist. I'm not going to learn to become one, and I'm definitely not a medicinal chemist. And you need all of that, plus really intrinsic, sort of focused knowledge in this area to be able to actually successfully push forward drug discovery. So I reasoned within the first few months, actually, of starting the lab that in addition to starting the lab, I wanted to develop an entity that would have its sole focus on this family of remodelers and extend it therefore to a number of other targets within the chromatin regulatory system with a new approach, a totally new approach to tackling chromatin biology, and approach that's very rooted in very rigorous basic mechanism and rooted in the biochemical mechanisms, because I think that underlies everything that we see at the cellular and ultimately tissue levels. And so with that, I had that idea and ultimately ended up partnering with Flagship Pioneering. My partner at that firm, Doug Cole, helped me start the company, and we were able to launch that company and officially launched the company in 2016. And just this past year, we took it public on NASDAQ. And so it's been very exciting, ride in developing both the lab really and the company. At the same time, I'm fortunate to work alongside such talented people and such wonderful leaders in the leadership of Foghorn that have really taken this company to new heights and, enabled us to get 3 programs spanning 2 different molecules in the clinic. One, an ATPase inhibitor that specifically targets the SMARCA4 and SMARCA2 ATPase engines of these remodelers that actually do the remodeling activity. That's being right now in 2 clinical trials, one in uveal melanoma and another one in acute myeloid leukemia, both enrolling actively. And then the second drug is actually a degrader for BRD9, which is a component of one of the forms of this complex I talked about. And that's being used and evaluated right now in the context of synovial sarcoma with the hope to be able to expand to other indications. And so this has been, a very exciting, I always say it's sort of a waltz between the lab and the company. And they're not overlapping in the sense that our lab is really exquisitely focused on unraveling all the basic mechanisms. And the company is, of course, very focused on drug discovery and a platform to enable that drug discovery. And it's been, very rewarding to be able to to participate in both and to drive this new class of therapeutics forward.
Foghorn has a very descriptive name for its platform called gene traffic control. What's meant by that?
Sure. So just as I described that, along our 2 meters of DNA, right, we have these some 20,000 odd genes, and we have to access those genes at the right times to be able to turn on specific programs. So just like when you're thinking about a taxiway at the airport on the tarmacs, you see a number of planes all geared up, and that sequence of timing of taking off and landing and moving around and ensuring that there are no crashes is very critical. And so our gene traffic control, that's essentially what these complexes do. They essentially dictate which genes are allowed to go, to take off, to turn on, and which genes are allowed to turn off or land, in the analogy of these planes.
Earlier you talked about having some terrific leaders. One of them is Adrian Gottschalk, your president and CEO. How did you connect with Adrian?
Yeah, so we are very fortunate to get Adrian, who's been an absolutely tremendous leader of this company, has such energy, such a passion for leadership itself, such a passion for company development, and just an amazing, batteries of skills from his previous experiences that we are very lucky to attract his attention to Foghorn. And it's been a privilege working with him ever since he first joined. And he had his very first meeting with me, now 3 or 4 years ago at Area 4 in Cambridge. We met to discuss just the overall idea and why I thought this was going to be a very exciting opportunity that's really, really fit for him. And it was really right after we started discussing this, it was clear that we wanted to work together. And shortly after that, he joined the company. So this is another advantage also of partnering with Flagship Pioneering. Their network is just so terrific that I was able to learn about and meet, and then ultimately even appoint people in the company, so many people that I otherwise wouldn't know from an academic perspective. So, having just been in the lab, I didn't quite know the bigger category of the pharma world or the biotech world. And so being able to have them as partners in building and developing the company was just invaluable.
You spoke of partners, and one that was announced last year was a collaboration with Merck and company, up to 425 million in potential milestones. What's been happening on that since last year?
Yeah, it's a really great partnership, and we're delighted to be partnering with Merck on those programs. This is for specific factors that we think interact with these chromatin remodelers, and so trying to go after that axis that is sort of one of the main axes actually that came from this rare disease setting I was talking about. This idea that complexes can be moved around in a way that they should or shouldn't be moved around on chromatin. We learned that from that rare disease setting with the fusion protein, dragging it to new sites, but turns out that the cell can use that in other ways as well. And transcription factors, for example, can bind and tether to certain surfaces of these remodeler complexes and actually help instruct where they're going on our 2 meters of DNA. They need instructive cues. Chromatin remodelers don't just sit on the genome at any old place. They have to have some guidance mechanisms. And so we've discovered that transcription factors play a major role in the guidance cues of these complexes. And so you can imagine, therefore, that especially given that transcription factors have been notoriously undruggable and a recalcitrant challenge for the field, just owing to the extremely high protein DNA affinities. These are picomolar affinities of the DNA binding domains of transcription factors to its DNA. That's been very, very hard to drug into. You need essentially a crowbar to try to pry those off of DNA in many cases. Well, whereas, these transcription factors, they don't act alone. They need other cofactors, and one of them actually is the BAF chromatin remodeling complex. And so those interactions tend to be much more weak and transient. And so we have a number of active efforts to try to go after those specific interactions.
Great. I know we have members of our audience with some questions and some additional questions from Juliana. Over to you.
Sure. Yeah, Toby, Shima has a question for you. So they say you have said a particular cancer might arise from more than one mutation. Since genetic treatment is based on dealing with one mutation as an antigen and developing antibodies against it, how much do multiple mutations in one cancer complicate genetic therapy?
Yeah, so, the way we think about it is that when you, for example, in pancreas cancer, you have, more than 90% of pancreas cancer patients have KRAS mutation. And about 70% have p53 mutation, some have p53 loss. The idea is that if you have a driver mutation, when you get rid of that mutation, typically, the cells are still addicted to it. It doesn't matter if they have other things. They're still addicted to it. So, they suffer. And there have been experiments done, in mouse models to actually show that when you get rid of KRAS, you get tumor regression. The tumor comes back, and there's a lot of other ways that the tumor comes back, but particularly, you do get a response. And so, by having therapies that are directed to the driver oncogenes, we think we can make, a really big dent on these tumors. Now, if you couple that with immune-directed therapies, then we think we can really eliminate the tumor altogether. And so that's the idea that we're pursuing.
Okay, great. Another audience question from Matt Segal. This is for you. Matt says, congrats on the Foghorn IPO. Do you see any extension of Foghorn into helping other diseases beyond cancer?
Yes, we do. In fact, I think, again, it comes from genetic evidence, but also some of the mechanistic evidence. So the other places, in addition to cancer, that we see these mutations are actually in neurodevelopmental syndromes, such as autism, intellectual disability. And then also, it's been increasingly shown that the activity of these complexes, which now Foghorn can target, is important in a number of cellular transitions, so cell fate decision-making, differentiation and maturation of cell types, as well as particularly is true in the immune system. So the immune system, T and B cells, require these complexes to achieve different states as they mature. And so we have definitely some goals in the immunology side as well as the neurobiology side. Thanks for the question.
Actually, I have a question for you, Segal, based on that. So one thing we've realized is that, we know the general principles of tumor formation, but we don't really understand that for metastasis. So what I mean by that is if you're like a strange person, you find a new tumor, you know to look for the driver mutation, so driver genetic alterations. If you find a metastasis,
Yep.
there are no, we can't really link metastasis to a particular driver mutation or genetic alteration. And what we're starting to understand now is that, actually metastasis is driven by a lot of epigenetic factors.
Right.
So are you thinking about this, targeting this in that space?
Yeah, absolutely. And I think to answer that question, I think really this is kind of going back to the academic side now. I think we have a lot more to understand. And it will come from model systems such as the ones you have, and increasing to try that across tumor types to really understand what is the role that these complexes can play specifically in the metastatic process. There are rare genomically well-characterized tumor types that do metastasize, synovial sarcoma being one of them, and there actually aren't clear recurrent events that genetically, right, are going to actually drive forward that metastasis. But what is different then still about the metastatic tumor versus the primary tumor, and that's, I think, a lot of work that we have yet to do. And how much would this complex and the activity of this remodeler, this critical determinant of DNA accessibility, play in that? I think there's a major play in that in the metastatic process. And I think we just need a bit more of a rigorous mechanistic understanding as to how that's occurring. It's a great question. We should talk more.
We'll do this again next month. So we only have one in our last minute. So the last question, and it has to be a little bit short, but if you could look in your crystal ball in the next year to 5 years, what do you think is going to be the next big, development that we'll hear so much about? Segal.
Sure. Well, I'm biased, but I would say that I think targeting master regulatory machinery that is, in charge of governing so many downstream pathways, be they signaling pathways, cell cycle pathways, apoptotic pathways, metabolic pathways, and others. I think, the crystal ball plus the hopeful crystal ball, I would say, is to be able to not only target them and succeed in targeting them and succeed in making them safe and effective, but increasingly with increasing specificity. So targeting specific activities, targeting specific targeting moieties, targeting specific roles that these complexes have that are important for one process or another, or just one aspect of that process, so that we can get better acuity about how to affect these tumor types and other diseases that are dependent on the activity of these regulators. I think there's a lot to be seen for the general field of chromatin biology / epigenetics. And I think, we're really just hitting the tip of the iceberg. There have been some enzymatic inhibitors, that have been developed over the years, but nothing that really comprehensively, affects the whole chromatin regulatory system. And I think, increasingly that's where we want to go. Broad, but also the ability to have focused therapeutics for each type of class and be able to take the data from sequencing and from other sources to be able to bucket patients and stratify patients based on who will respond based on what kind of mutation they have, irrespective of the tumor type, oftentimes.
Yeah, I'm gonna follow that up with my bias also, and I think in the next 5 to 10 years for sure we'll have new immunotherapies that not only drive T cells to kill cancer cells, but NK cells, and you're starting to see some of that now with CAR NK cells. So therapies targeting NK cells. Therapies targeting innate immune cells such as macrophages and all. And a combination of this will really be very important in the next couple of years. 2 more things. One thing we're really excited about is, this small molecule inhibitors of like big oncogenes such as KRAS. KRAS G12C, is basically been taken down. Very soon you're going to have inhibitors against KRAS G12D and G12V. And finally, I'm really excited about this molecular glues and degraders and the ability to really use this to target the undruggables.
All right, terrific. Well, that brings us to the end of this session of GenLive. I want to again thank Sigal and Toby for a terrific discussion, and we look forward to following all of your work in this area. Thanks also to Anjali, Alex, and the rest of the Gen production team for making this episode run so smoothly. And thanks again to Isoplexis, our sponsor. And of course, thanks to all of you for tuning in. Don't miss our next GenLive in October, which will be a celebration of Gen's 40th anniversary. To celebrate, John Sterling, our editor-in-chief, will convene a celebration covering the twists and turns of the biotech industry over the last 40 years with some old friends and special guests. I'm Juliana Lemure. Stay safe, and we'll see you next month.
Thank you, everybody. Great to see you.
Thank you.